Morning, everyone. I hope you can hear me. Can you hear me?
Yes, good morning.
Okay, thanks. Great. We can wait a few more minutes for the others, but before we start, I have a question for those people who were waiting in the waiting room. How did you join to the meeting? Yeah, I know, but you did it directly from MS Teams, yes? Through the this molecular diagnostics group that I have created. Okay.
No, I joined through the code number.
Okay.
It doesn't add me to the group. It just allows me to come in.
Okay, so I had to accept your you to join here. My name is Magda Rybicka-Misiejko, and for those who don't know me, I work in the laboratory of photobiology and molecular diagnostics, and in the laboratory of genetic analysis. And I would like to welcome you at the molecular diagnostics course that is composed of two main parts. The first one are lectures, and usually we give 12 to 15 lectures. And the second part is a training course that is starting on November. This year we will have 6 meetings in real time, so you will have the opportunity to work a little bit in the lab, and I hope you will enjoy it. The final exam covers all the subjects that were mentioned or discussed during both parts of molecular diagnostic course. And today, I would like to tell you a little bit about the introduction. I will give you just an introduction to molecular diagnostic.
I'm sorry, did you mean to mute yourself?
Could you please repeat, because I did not hear?
You've become muted, and I asked if it was on purpose.
Yes, it was, because I cannot show you the presentation because this laptop is quite new, and actually I should do this earlier. I don't know what is going on, but just give me 2 minutes.
Sure.
What seemed to be problem? Maybe we can help.
Okay. Okay, thank you. So, introduction to molecular diagnostics. But I cannot change. Okay. So today I will mention you about the essential concepts in molecular diagnostics that may impact on the identification of markers of human diseases. I will present different types of clinical genetic testing that are currently used in molecular diagnostics lab. Also explain a little bit steps of developing molecular genetic tests from research lab, and present you the international standards that lie behind the principles of analytical validity, clinical validity, and clinical utility when using genetic testing techniques. And I will also describe you in brief some examples of different molecular diagnostics platforms. Of course, molecular diagnostics would not be possible without the many significant pioneering efforts in genetics and also molecular biology. Earlier observations in genetics began with the discovery of the inheritance of biological traits made by Mendel, and it was in 1866, and the observation in 1910 that genes were associated with chromosomes by Morgan. Later in 1949, Pauling and his co-workers introduced the term molecular disease in the medical vocabulary, and it was based on their discovery that a single amino acid change at the big globin chain leads to sickle cell anemia, characterized mainly by recurrent episodes of acute pain due to vessel occlusion. And the seeds of molecular diagnostics begins from the first description of the structure of DNA by Watson and Crick, of course, and it was, as you probably know, in 1953. And the description of the DNA structure initiated the huge increase in the knowledge of the biology and chemistry of our genetic machinery. And the impact of the Watson and Crick discovery was so significant that it is now considered one of the most important scientific discoveries of the 20th century. And why it was so important? Because Watson and Crick, they did not only describe the structure of DNA, but they also hypothesized about many of its properties, which took decades to confirm. And one of those properties was the replication of DNA, which was shown to be semi-conservative. And at the same time, DNA polymerase that replicates the DNA was discovered, and identification of the genetic code was important for understanding the information stored in DNA. Additional studies described the transcription and translation processes and uncovered several startling findings. Along with the discovery of the basic biology of genes and their expression, many other important techniques were invented and the Southern blot methods, which identified specific electrophoretically separated pieces of DNA. This method participated in many discoveries and was one of the first molecular diagnostics methods to be used to test for genetic diseases. Of course, DNA sequencing technologies were invented later, and further advances in these technologies led to the first large biological science research undertaking, the Human Genome Project. You probably have heard about it. And along with DNA sequencing, further technical discoveries, including the polymerase chain reaction and microarray technology, became methodologic foundations of the molecular diagnostics. Molecular diagnostics is a broad term describing a class of diagnostic tests that assess a person's health at a molecular level, detecting and measuring specific genes, genetic sequences in DNA or RNA or the proteins they express. Molecular diagnostics identify gene, RNA, and protein variations that shed light on whether a specific person is predisposed to have a disease, whether they actually have a disease, or whether a certain treatment option is likely to be effective for a specific disease. And these tests also can detect and quantify the presence of specific viruses, bacteria, or types of cells. Nowadays, more than 80% of molecular tests performed address infectious diseases detection and management. The test menu for infectious diseases include human immunodeficiency virus, so HIV, hepatitis B virus, HBV, hepatitis C virus, HCV, and many others. Other molecular tests are applied in forensic medicine, paternity testing, tissue typing, oncology, and food testing. The molecular diagnostic testing can be performed on very small amounts of tissue obtained from small biopsies, but testing can even be performed on archival material, so material that has been collected in the past and fixed in formalin. What is very important, it is usually not necessary to take extra tissue for these tests, since most of them can be performed on tissue that is left over after routine diagnostic analysis. Molecular diagnostics is becoming increasingly important as an essential tool for the rapid, accurate, and cost-effective delivery of safe and effective therapy for many diseases. And its uptake is driven by continuous innovation in underlying technologies, constant identification of new or improved biomarkers, steady disease in associated costs, and increasing automation that enables even complex assays to be performed. Tests may be relatively unsophisticated, such as those used for the detection of pathogen-specific genomic sequences. They may involve the identification of complex disease-associated mutations and also epigenetic changes in DNA sequences, or different expression patterns of RNA and proteins. They may also allow for the identification a predisposition to a particular disease development. The information provided by genomic sequencing is critical for our understanding of the interactions, which determine an individual's health at a molecular level. And whereas the first draft sequence of the human genome took 10 years of worldwide efforts, it is now possible to sequence a complete human genome in less than 1 day. Now, this is beginning to allow the identification of every individual's unique molecular characteristics and to apply them to personalized diagnosis, treatment, and susceptibility prediction. And continuous innovation in technology is increasing the speed and performance of molecular diagnostics, and the future in which whole genome sequencing is routinely performed is not that far away. Diagnostics are an integral and critical part of our healthcare system. As the results of these tests inform a wide range of medical decision-making, from the genetic tests that inform personalized cancer treatment to the microbial culture that identifies the right antibiotic to fight an infection. Diagnostic tests provide critical insights at every stage of medical care. As you can see on this slide, it is included in prevention, detection, diagnosis, treatment, and successful management of health conditions. Gene-based molecular diagnostics is changing the practice of medicine and will continue to do so far for the nearest future. The primary molecular diagnostics approaches are basically based on two major database sciences, genomics and proteomics, and their relationship to disease and metabolic processes, so functional genomics and functional proteomics. The genome or proteome that forms the basis for these tests is very often of human origins, but may be also from a pathogen. As a result, the range of application for molecular diagnostics is actually unlimited. Diagnostic testing is used to identify or rule out a specific genetic or chromosomal condition. In many cases, genetic testing is used to confirm a diagnosis when a particular condition is suspected based on physical signs and symptoms. Newborn screening is used just after birth to identify genetic disorders that can be treated early in life. Prenatal testing is used to detect changes in fetus genes or chromosomes before birth. This type of testing is offered during pregnancy if there is an increased risk that the baby will have a genetic or chromosomal disorder. Carrier testing is used to identify people who carry one copy of a gene mutation that, when it's present in two copies, causes a genetic disorder. There are also pre-implementation testing that are also called pre-implementation genetic diagnosis. This is a specialized techniques that can reduce the risk of having a child with a particular genetic or chromosomal disorder. It's used to detect genetic changes in embryos that were created using assisted reproductive techniques, such as in vitro fertilization. Predictive and presymptomatic types of testing are used to detect gene mutations associated with disorders that appear after birth, often later in life. in life. These tests can be helpful to people who have a family member with a genetic disorder, but who have no features of the disorder themselves at the time of testing. And the last type is forensic testing that uses DNA sequences to identify an individual for legal purposes. Unlike the tests described earlier, forensic testing is not used to detect gene mutations associated with disease, and this type of testing can identify crime or catastrophe victims, rule out or implicate a crime suspect, or establish biological relationship between people, for example, paternity testing. In medicine, in general, a biomarker is a metric characteristic that reflects the severity or presence of some disease state. More generally, a biomarker is anything that can be used as an indicator of a particular disease state or some other physiological state of an organism. And in 2001, the term biomarker was defined by a working group of the National Institutes of Health as a characteristic that is objectively measured and evaluated as an indicator of normal biological processes, pathogenic processes, or pharmacological responses to a therapeutic intervention. And biomarkers include genes, proteins, genetic variations, and differences in metabolic expression from different sources, such as body fluids or tissues. In clinical trials, biomarkers can be used to create subgroups that are used in the stratification of study subjects. So the utilities of biomarkers include identifying patients with a good prognosis versus poor prognosis in the absence of medical treatment, or identifying patients that will likely to respond to a given drug versus patients unlikely to respond to the drug, or identifying patients likely to experience adverse drug reactions to a given drug versus those that will not experience these adverse drug reactions. A biomarker can be a substance that is introduced into an organism as a means to examine organ function or other aspects of health. It can be a substance whose detection indicates a particular disease state. For example, the presence of an antibody may indicate an infection. More specifically, a biomarker indicates a change in the expression or state of a protein that correlates with the risk for or progression of a disease or with the susceptibility to the disease. Biomarkers are characteristic biological properties that can be detected and measured in parts of the body, in blood or tissues. They may indicate either normal or disease processes in the body. Biomarkers can be classified based on different parameters, including their characteristics, such as imaging biomarkers or molecular biomarkers. Molecular biomarkers can be used to refer to non-imaging biomarkers that have biophysical properties, which allow them to be measured in biological samples and include nucleic acid-based biomarkers such as gene mutations or polymorphism and quantitative gene expression analysis, peptides, proteins, lipids, metabolites, and other small molecules. Biomarkers can also be classified based on their application such as diagnostic biomarkers, staging of disease biomarkers, disease prognosis biomarkers, and biomarkers for monitoring the clinical response to an intervention. Another category of biomarkers includes those used in decision making during early drug development. Biomarker requirements for chronic diseases whose treatment may require patients to take medications for years. Accurate diagnosis is particularly important, especially when strong side effects of the treatment are expected. And in these cases, biomarkers are becoming now more and more important because they can confirm a difficult diagnosis or even make it possible in the first place. A number of diseases, and in particular chronic disorders, often begin with an early symptom-free phase. And such symptom-free patients may have more or less probability of developing symptoms. In these cases, biomarkers help to identify these high-risk individuals so that they can either be treated before the onset of the disease or as soon as possible thereafter. And to use a biomarker for diagnostics, the sample material must be as easy to obtain as possible. This may be a blood sample taken by a doctor, or a urine or saliva sample, or a drop of blood like those diabetes patients extract from their own fingertips for regular blood glucose monitoring. The speed with which a result is obtained from the marker test is critical for rapid identification of treatment. A rapid test that delivers a result after only a few minutes is optimal. This makes it possible for the physician to discuss with the patient how to proceed, and if necessary, to start treatment immediately after the test. However, a predictive biomarker assay for patient stratification should include an in vitro diagnostic test, which requires intensive clinical and validation steps. Of course, the method used to detect a biomarker must be as easy as possible to carry out. The results from different labs may not differ significantly from each other, and the biomarker must have proven its effectiveness for the diagnosis, prognosis, and risk assessment of affected diseases, of course, in independent studies. Biomarkers' journey from discovery to clinical use is long and complicated, but it can be broken into phases or steps. Biomarker discovery efforts have increased with the emergence of technologies for gathering relevant data. For example, single-cell next-generation sequencing, liquid biopsy for circulating tumor DNA. Liquid biopsy is just a blood sample, as you probably know. Microbiomics, radiomics, and other types of high-throughput technologies have exploded in popularity in recent years. Biomarker development can be described as being divided into three phases: discovery, verification, and validation. And the validation phase is often divided into two stages: analytical validation and clinical validation. And the clinical validation is often described as qualification. Biomarker discovery aims to identify a potential biomarker using available specimens. And in this step, randomization and blinding can be used to avoid bias. This step uses also appropriate analysis method and control for multiple testing and considers the levels of evidence. The intended use of a biomarker, for example, the risk stratification and screening, and the target population to be tested need to be defined early in the development process. The use of a biomarker in relation to the course of a disease in specific clinical context should also be pre-specified. The patients and specimens should both directly reflect the target population and intended use. Bias, bias is a systematic shift from true, is one of the greatest causes of failure in biomarker validation studies. Bias can enter a study during patient selection, specimen collection, specimen analysis, and patient evaluation. Randomization and blinding are two of the most important tools for avoiding this bias. Randomization in biomarker discovery should be carried out to control for non-biological experimental effects owing to changes in reagents, technicians, equipment, and so on, that can result in batch effects. Specimens from controls and cases should be assigned to arrays, testing plates, or batches by random assignment, ensuring the distribution of cases, controls, and age of specimen are equally distributed. Blinding can be carried out by keeping the individuals who generate the biomarker data from knowing the clinical outcome. And this prevents the bias induced by unequal assessment of biomarker results. Randomization and blinding should be used in the process of biomarker data generation, and these two steps should be incorporated at every stage of a study when it is possible. Analytical methods should be chosen to address study-specific goals and hypotheses. Data-driven analysis and the resulting findings are less likely to be reproducible in an independent set of data. So the analytical plan should be written and agreed on by all members of the research team before receiving data to avoid the data influencing and analysis. This includes defining the outcomes of interest, hypotheses that will be tested, and criteria for the success. And control of multiple comparisons should be implemented when multiple biomarkers are evaluated. A measure of false discovery rate is especially useful when using large-scale genomic or other high-dimensional data for biomarker discovery. What's more, during biomarker discovery, evaluation of associations between a biomarker and disease status or clinical characteristics such as age, gender, and body mass index, or in diseased patient stage or other disease characteristics, can inform design of future validation studies. Analytical validation aims to establish the performance characteristics of a biomarker, including sensitivity, specificity, precision, and other relevant performance characteristics following a pre-specified protocol. The statistical analysis methods used for analytical validation are similar to those that are used to biomarker discovery, and the goal of analytical validation is to reveal a biomarker's technical performance. For example, the biomarker will provide consistent measurements to the unknown true values and not its usefulness. Clinical validation aims to establish an association between the biomarker and the endpoint of interest, and to reveal the usefulness of the biomarker. And this clinical validation relies on external validation and can be done by retrospective use of clinical trial data or by prospective clinical trials. Retrospective use of clinical trial data is a form of external clinical validation in which the biomarker evaluation is not part of the original study design. To demonstrate clinical utility, it is necessary to establish the clinical usefulness of a biomarker. For example, whether the use of the marker in clinical care results in improved patient outcomes. Retrospective use of prospectively collected specimens, for example, the probe design is appropriate for validating diagnosis screening and prognostic biomarkers. Generally, 4 factors are considered when evaluating the clinical utility. Patients' outcomes, so do the results of the test improve health outcomes, so reduce mortality, increase patients' quality of life, diagnostic thinking, decision-making guidance, or familial and societal impacts. So there are many questions that scientists need to find the answer before the end of the discovery of a molecular biomarker. There are many genomics and proteomics techniques are available for biomarker discovery. Few recently used are shown here. Genomic approach has been extensively used for biomarker discovery and identification, with approximately 30,000 genes in our genome, realizing the clinical potential of genome scale information requires better methods of viewing, analyzing, and utilizing the complex large-scale biomarker data. But the new platform technologies such as deep and high-throughput sequencing enable researchers and scientists to make great progress recently. Sometimes proteomics, even more than gene expression analysis, is an important tool in understanding the complexities of human physiology and the state of disease. Proteomics has potential for biomarker discovery, especially that of novel diagnostic and predictive biomarkers. Proteomic techniques include Western blots, staining, ELISA, and mass spectrometry. Secretomics is a subfield of proteomics that studies secreted proteins and secretion pathways using proteomic approaches. And this subfield has recently emerged as an important tool for the discovery of biomarkers. In what is now commonly referred to as proteogenomics, proteomic technologies are further used for improving gene annotations. Metabolic approach has recently been introduced to address the global analysis of all metabolites in a biological sample. Related term metabonomics was introduced to refer specifically to the analysis of metabolic responses to drugs or diseases. Tissue-based biomarkers, they are not shown here, but they are one of the best established biomarker in research areas. Almost no cancer treatments are initiated without a diagnosis of the cancer itself using either conventional methods or molecular tests. A cancer biomarker usually refers to a substance or process that is specific for the presence of cancer in the body. A biomarker, as I mentioned before, may be a molecule secreted by a tumor or a specific response of the body to the presence of a cancer. A number of techniques are employed in modern diagnostics to detect and quantify specific DNA or RNA sequences. These are only the most important ones. I will briefly tell you about them. So the most important polymerase chain reaction is one of the most essential methods underlying many molecular diagnostics diseases. This is the process of making copies of a specific DNA or RNA sequence that may be found in a sample until there are so many copies that they can be detected and measured. There are a number of different amplification technologies, but PCR reaction is the most widely used still and is considered a workhorse in molecular diagnostics. PCR already has very widespread applications, and new uses are being devised on a regular basis. PCR can amplify a single DNA molecule from a complex mixture, largely avoiding the need to use DNA cloning to prepare that molecule. There are variants of the technique that can similarly amplify a specific single RNA molecule from a complex mixture. DNA sequencing has been greatly simplified using PCR, and this application is now very common. By using specific primers, it is possible to use PCR to create point mutations, deletions, and insertions of target DNA, which greatly facilitates the analysis of gene expression and function. PCR is very sensitive and can amplify small amounts of DNA. So using appropriate primers, very small amounts of specified bacteria and viruses can be detected in tissue, making PCR invaluable for medical diagnosis. PCR is now important for characterizing medically important DNA samples. PCR is now very important also in forensic medicine. It's even possible to use PCR to amplify the DNA from a single human hair or microscopic drop of blood left at the scene of crime to allow detailed characterization. Real-time PCR is a technique that's used to monitor the progress of a PCR reaction in real time. At the same time, a relatively small amount of PCR product can be quantified. It's based on the detection of the fluorescence produced by a reporter molecule, which increases as the reaction proceeds. It's also known as a quantitative polymerase chain reaction, which is based on the traditional polymerase chain reaction. It has many advantages over the normal PCR reaction. It gives a look into the reaction that is help to decide whether reactions have worked well and which have failed. The efficiency of the reaction can be calculated. There is no need to run the PCR product out on a gel after the reaction, as the melting curve analysis serves this purpose. And the real-time PCR data can be used to perform quantitative analysis of gene expression. It is also faster than normal PCR and less complex. Real-time PCR allows the success of multiple PCR reactions to be determined after only a few cycles, without separate analysis of each reaction, and avoids the problems of false negatives. Another method, in situ hybridization, is a type of hybridization that uses a labeled complementary DNA, RNA, or modified nucleic acid strand to localize a specific DNA or RNA sequence in a portion of a section of tissue, or if the tissue is small enough, in the entire tissue, in cells, and in circulating tumor cells. FISH is an effective technique that enables direct visualization of genetic alterations in the cell. This technique has many applications and is generally used to examine either imbalances, loss of segments of chromosomal materials, or to show specific breakpoints with or without imbalance. This method was initially used for classification of chromosomes, but this technique has since been adopted in a range of applications in the medical and biological fields. Common uses of FISH are chromosomal gene mapping, characterizing genetic abnormalities, identify genetic abnormalities related to genetic diseases, and even detecting viral genomes. DNA microarray is a collection of microscopic DNA sequences, oligos, that are attached to a solid surface. These sequences represent fractions of a large library of genes that are present in a cell. If a gene is active within a cell, then the DNA will bind to its complementary oligo. If the DNA has been fluorescently labeled, then the complementary oligo can be identified. Each oligo represents a known gene sequence, so genetic diseases are identified according to which oligos are hybridized. The greater the expression of a particular gene, the brighter the fluorescence seen in the relevant microarray well. In addition to diagnosing genetic diseases, microarrays can also be used to identify new genes that cause disease conditions. If DNA from healthy and diseased cells are labeled with different fluorophores, comparison of gene expression can be made. Genes that are only active in a diseased or normal state will be particular of interest to scientists. And when microarrays are used to compare mRNA expression in healthy and diseased cells, the following conventions are used. DNA from healthy cells are usually tagged with green fluorescence, while DNA from diseased cells are tagged with red. And this enables for identification of genes present only in healthy cells dots, diseased cells, red dots, both yellow and neither gray. Another method, mass spectrometry. This is a technology that determines the molecular mass of charged particle by measuring its mass to charge ratio. This technology is used to find and analyze protein-based biomarkers and also nucleic acids-based biomarkers, and is broadly classified into gel-based and gel-free techniques. Mass spectrometry is used to quantify known materials, to identify unknown compounds within a sample, and to elucidate the structure and chemical properties of different molecules. Mass spectrometry basically studies the effect of ionizing energy on molecules. It depends upon chemical reactions in the gut phase in which sample molecules are consumed during the formation of ionic and neutral species. The first step in the mass spectrometric analysis of compounds is the production of gas phase ions of the compound, basically by electron ionization. This molecular ion undergoes fragmentation. Each primary product ion derived from the molecular ion undergoes fragmentation. The ions are separated in the mass spectrometer according to their mass to charge ratio, and the mass spectrum of the molecule is produced, and it displays the result in the form of a plot of ion abundance versus mass to charge ratio. Ions provide informations concerning the nature and the structure of their precursor molecule. Sanger sequencing, it's a targeted sequencing technique that uses oligonucleotide primers to seek out specific DNA regions. It begins with the denaturation of double-stranded DNA into two single-stranded DNA fragments. Next, a fragment of single-stranded DNA is multiplied into millions of copies. Next primer that corresponds to one end of the treatment is attached, and the fragments are then added to 4 polymerase solutions. Each solution contains the 4 types of bases, but only one type of terminating nucleotide. And the chain grows until a termination nucleotide is randomly added, and the resulting double-stranded DNA fragments are denatured to obtain a series of single-stranded DNA of different length, and the fragments are then separated by electrophoresis, and the sequence may be read. And the next generation sequencing is a powerful method that has enabled the sequencing of thousands of millions of DNA molecules at the same time. Next generation sequencing is also known as high throughput sequences, and it describes a number of different modern sequencing technologies. The high demand of low-cost sequencing has driven the development of high throughput sequencing, which produce thousands or millions of sequences at once. They are intended to lower the cost of DNA sequencing beyond what is possible with standard sequencing methods. So these recent technologies allow to sequence DNA and RNA much more quickly, and cheaply than the previously used Sanger sequencing, and such have revolutionized the study of genomics and molecular biology. Next generation sequencing is not a single technology, but rather several different technologies that share a common feature of massively parallel sequencing of amplified or single DNA molecules in a flow cell or chip. And next generation sequencing can generate in a single instrument run hundreds of millions to gigabases of nucleotide sequences data, depending upon platform configuration, chemistry, and flow cell or chip capacity. And now some examples of successful molecular diagnostics. I think the most successful treatment of cervical cancer is delivered as a result of early detection. The use of human papilloma virus tests to identify women most likely to develop cervical cancer has the potential to revolutionize treatment of this. About 20 years ago, researchers discovered a link between the human papilloma virus, HPV, and cervical cancer. It is known that of the 100 of all genetically distinct types of HPV, about 14 are associated with a high risk of cervical cancer, and just 8 of those are responsible for almost all cases. And because HPV types can be genetically differentiated, it has been possible to develop molecular diagnostic tests to screen women for the high-risk types. And identifying women with these high-risk types enables those women to schedule more frequent exams and can guide a physician to follow up more often and more aggressively when pap tests yield abnormal or inconclusive result. In this way, molecular diagnostics are driving the next advances in early detection and prevention of cervical cancer. The cystic fibrosis test is an example of an molecular diagnostic test that can be classified as a screening test or a diagnostic test, depending on when it's used. Cystic fibrosis is an inherited chronic disease that mainly affects the lungs and digestive system, and currently affects about 70,000 children and adults worldwide, and about 1,000 new cases are diagnosed each year. And cystic fibrosis is caused by an abnormality in the CF transmembrane regulator, so CFTR gene, that prevents the normal movement of chloride ions across membrane. Over time, patients have serious breathing and digestive difficulties, and mean life expectancy is less than 40 years. So cystic fibrosis is seen in patients with 2 altered CFTR genes, one from each parent. Those that only have 1 copy of the altered gene are considered as carriers. And while they do not have symptoms of cystic fibrosis, they can pass the gene to their children. And therefore, carrier screening of parents to be or pregnant women determines their CFTR gene status and helps families adequately prepare for the result. One of the most common mutations causing cystic fibrosis is the delta F508 deletion mutation, which can be detected using a number of molecular techniques. These tests use DNA amplification methods to amplify key portions of CFTR gene and look for the mutations that cause cystic fibrosis. An example of molecular diagnostic that improves therapeutic decision making is KRAS testing. KRAS is a protein encoded by the KRAS gene that plays a critical role in cell division, cell differentiation, and self-destruction of cells. And KRAS mutation that produce an abnormal overactive KRAS protein are found in many different cancers. Detecting KRAS mutation is used to determine patients' suitability for certain therapies in colorectal and lung cancer nowadays. Anti-epidermal growth factor receptor drugs are now key therapies for those cancers, but patients whose cancers contain KRAS mutations usually fail to respond to these therapies. And KRAS mutation is typically performed for patients via direct DNA sequencing or PCR or other amplification-based methods. KRAS mutation testing is just one of a growing number of examples of molecular diagnostics that help inform a physician's decision-making regarding appropriate treatment. Identifying which patients are unlikely to respond to a specific kind of therapy reduces the costs of ineffective treatment and increases also patients' quality of life very often. Finally, many molecular diagnostics improve monitoring and management of diseases. Some of the most common monitoring molecular tests are tests for HIV viral load in patients affected with this virus. HIV viral load tests are amplification-based tests that measure the amount of HIV RNA to determine how many copies of the virus are present. Keeping the viral levels as low as possible is key to staying healthy with HIV infection. Because researchers and physicians have made great progress in the care of HIV patients in the 3 decades since HIV was first detected. The HIV viral load test has become an essential tool as patients cycle through different antiretroviral therapies and develop resistance to certain therapies. And periodic HIV viral load tests indicate whether or not the disease is progressing, either because an untreated patient's immune system can no longer suppress replication of the virus, or because the therapy for a patient in treatment is no longer working. The HIV viral load test is not used for initial diagnosis. This is with antibody tests, but typically also is used upon initial diagnosis as a staging diagnostic to get a first report on the progression of the disease prior to treatment. Future perspectives. So this lecture includes only a few of hundreds of examples in which the latest molecular technology is being used for rapid diagnosis and to aid physicians in improving clinical outcomes for their for their patients. Since we are only in the early stages of understanding the complex fields of genomics and proteomics. However, diagnostic technologies are evolving rapidly. It seems fair to say that much of the promise of molecular diagnostics is still ahead of us. As we understand more about the complexity of diseases, there are an increasing number of molecular biomarkers that must be identified to fully characterize the many subsets of populations that have highly variable symptoms and clinical courses. New technologies are making it possible to gather more information to more fully characterize the disease's states. The best treatment alternatives for patients, or in the case of infectious diseases, rapidly identify a specific pathogens of many. Molecular diagnostics are truly transformative technologies, but yet the excitement about the future of this field must be a little bit tempered by recognition of the challenges of continuing innovation and timely patient access. The challenges include regulatory hurdles and practical and ethical issues that are associated with generating and storing large volumes of highly detailed and personal health information. But the fast pace of technological development in molecular diagnostics and the rapidly growing body of research regarding biomarkers challenge regulatory bodies to stay current and clear understanding of regulatory expectations for multiplex tests and next generation sequencing, as well as the increasing investments necessary to generate the clinical evidence required for approval or clearance will be integral. That's all what I have to say for today. It was quite, I think, intense, but I was trying to cover all the subjects that will be mentioned during all the lectures. So if you have any questions, you can ask me now. If not.
Excuse me, which textbooks do you recommend to prepare for exam?
Textbook. I do not really. I don't have one that I can recommend. I can prepare some review papers for you for the next exam, because usually you should search for the information by your own, but I can give you some examples of the books or review manuscripts.
Excuse me, our lectures are in English. Does that mean that we will have exam in English as well?
Yes, it will be in English.
Okay,
Because
thank you.
I think no, the lectures are in English, but the practical courses, I think 5 groups are in Polish and 1 is in English. But the exam will be in English.
Thank you.
Will you share these presentations as a PDF or PowerPoint files?
I'm not sure yet, but usually I do not share. Because there is a lot of work to prepare this presentation. Okay, if there is no more questions, then thank you for your attention and see you next week. Goodbye.
Thank you.
Thank you.
Bye.
Bye.